Hey everyone, I have a really special paper today. Maybe I always say that, but I promise this one is really good. It has to do with genetic circuits in yeast. So building this actual genetic circuitry in yeast, and it's a cool yeast too. This paper here was published in the journal Metabolic Engineering, which is, for this field, sort of the top or maybe second from top best journal you can get into. And the title here is Repurposing Plant Hormone Receptors as Chemically Inducible Genetic Switches for Dynamic Regulation in Yeast. So plant hormone receptors refer to Pure 1 biosensors, which I've talked about in other videos, so those are really cool on their own. Chemically inducible genetic switches, that sounds really cool, and that's because it is really, really awesome. So we're dealing with using these Pure 1 biosensors instead of as biosensors as inducible, so chemically inducible, meaning we add a chemical and we turn on a particular gene or we turn off a particular gene with that chemical. So chemically inducible genetic switches for dynamic regulation, so changing regulation over time. So this is, the genetic circuitry part of this paper in yeast. And specifically, we're working with a non-conventional yeast called Kluyveromyces marxianus, and we'll talk about that in a second. So I am one of the authors listed on this paper, but I'm sort of lost in the middle, so I contributed very little to this paper. Typically in these journal articles, the author who contributed the most but is not API is listed first. So this is Shuang Wei. She now works in industry, but she was a PhD student in my lab. Mary or Mengguan Li, she was also a student in my lab. She got her PhD same time as Shuang. Jie was a postdoc in my lab, overlapped with me just a little bit. And then there's me. And then there's a research scientist, Sang-Yul Park, who basically discovered the Pure 1 biosensors in Sean Cutler's group. And then Ian Wielden is my PI and the PI of all of my lab mates at the beginning of this paper. All right. So this is a metabolic engineering article, right? We're doing engineering here. We're not doing fundamental biology. So anyway, let's start with the abstract. As always, go through the introduction a little bit and then go through all the figures and really figure out what's going on. So here we're talking about precise control of gene expression. So when you're doing metabolic engineering, you want to produce some chemical in a microorganism typically. You add all of these genes, and then you use this metabolic pathway to make the chemical you want. And you may think it's as simple as just slapping those genes in and pressing play, and then your organism makes what you want. But there's all of these nuances of how much do you turn on particular genes, and how much do you divert energy away from necessary cellular functions, right? These microorganisms have evolved millions of years to be, really billions of years to be optimized, right? So they don't have extra resources. They're using all of the resources really that they have in order to survive, right? So typically when we do metabolic engineering, we have these hardwired approaches to pathway engineering, meaning we just add these genes, they're on all the time, right? But what if we could change the regulation of those genes over time? So that's a lot of what this paper has to do with. Again, we're working in this non-conventional host, Kluyveromyces marxianus, and we're going to be using a 2-channel chemically regulated gene expression system. So this is some of the genetic circuitry. 2-channel, meaning we have 2 tunable knobs in this yeast. Genetic circuits is a really big field, and in E. coli, I know there's papers that use dozens of biosensors all at once, control dozens of genes all at once, have all of these complex and OR gates built in a microorganism. But here we're just demonstrating a 2-channel system, so just 2 knobs that we can turn with chemical regulation in order to change the gene expression. So Kluyveromyces marxianus is a non-conventional yeast. It is interesting in a lot of ways. It's my favorite yeast. When you grow it up, it smells like a Hefeweizen style beer, if you've ever had one of those before, because it makes some small molecules like 2-phenylethanol, which smells like roses, and ethyl acetate, which we'll be talking about in this paper. It grows about twice as fast as other yeasts that are commonly worked with, which is great. That means all of your experiments go about twice as fast. And in industry, of course, time is money. And it can grow at high temperatures, so it's a thermotolerant yeast. It can grow around 50 degrees Celsius or even higher in some cases, which is also industrially very relevant. Again, we're dealing with these Pure 1 biosensors, but we'll also talk about these sort of modified Pure 1 biosensors. And our goal here is to redirect metabolic flux. So flux, again, is that idea that we can push mass or moles of a compound through a metabolic pathway that we want. Right, so we may want to divert some of the cell's energy into growing more cells and some of that cell's energy into making our product. And we're doing that here by regulating two genes. One is called pyruvate dehydrogenase, or PDA1, and the other is pyruvate decarboxylase, or PDC1. So we'll see these a lot in this paper. And again, we're chemically tuning gene expression, not just in K. marxianus, but first we demonstrated the system in Saccharomyces cerevisiae, which is baker's or brewer's yeast. It's the most well-studied yeast. It's the conventional yeast, if you want to call it that. The beginning of this introduction has a pretty good definition of metabolic engineering. Metabolic engineering seeks to harness native and heterologous pathways for the high titer, rate, and yield production of chemicals. So this is saying native pathways are pathways already in the yeast. Heterologous pathways are pulling yeast or pulling genes from other organisms and putting them in yeast. So for example, you can go back and see the psilocybin production in yeast review video, or the cannabinoid production in yeast, or opioid production in yeast. In those cases, we're taking heterologous pathways, so genetic pathways from other organisms, and putting them in yeast. So those are heterologous from other organisms. And the purpose is for increasing basically the production of the chemical that we want, right? So this is a definition of metabolic engineering. So often in industrial settings, if you're trying to produce a product, the most advantageous thing to do is to, what it says here, separate growth and production phases, growth and product production phases. So you start a culture, you have very few cells. In the beginning, you want to make a lot of cells. You want to make a lot of your microscopic factories. You make a lot of them, and then you want to turn on production of the chemical that you want, because often production of the chemical that you want is detrimental to the cell growth. So there's this middle ground of you want to have a lot of cells that are producing your compound, and then you want them to produce your product. You don't want them to be stressed out producing your product in the beginning and not making more of the factories for you. That's not optimal. And you solve this problem with something called dynamic regulation. So changing the regulation of genes over time. That's the dynamic part, over time. Next, we're talking here about the Pure 1 biosensor system. Here we're using it as a metabolic switch, or a valve, potentially you could call it. Pure 1 ABA, or Pure 1 abscisic acid. ABA is abscisic acid. This is wild type Pure 1. This is the normal Pure 1, but we're using it in yeast. So we're using Pure 1, this Pure 1 Hab1 system, where Pure 1 is this biosensor that detects abscisic acid. Once it detects abscisic acid, then Pure 1 undergoes a conformational shift, so it moves slightly and it can bind to this other protein called Hab1. And so this dimerization, this putting together of these two molecules, allows us to control gene expression. So Pure 1 is advantageous because it has this malleable binding pocket. So where that chemical fits can be modified, and I talked about this a lot in other videos. That Pure 1 binding pocket can be modified to detect other chemicals. And here we're showing that this system can be made into a similar orthogonal system. So orthogonal meaning two things or multiple things happening at the same time. So we'll be talking about Pure 1 star Mandy. So Mandy is short for mandipropamid. It's just agrochemical that we happen to have a Pure 1 biosensor for. And this star thing is really important. So normally you can think of Pure 1 and Hab1 as sort of like magnets where opposites attract, because what happens is once Pure 1 binds to a chemical, it can then undergo an ionic bond between a couple of amino acids on Pure 1 and Hab1 that form a salt bridge between the two proteins. So this is a really, really strong bond between these two proteins, and it sort of locks in the chemical that it's sensing. So that effectively raises the sensitivity of the entire system as a whole, but that's some molecular dynamics kind of stuff we don't care about here. But the idea here, you can think of pure-1 as like positively charged, for example, and Hab1 as negatively charged. So pure-1 and Hab1 can stick together, right? But you can actually swap the salt bridge so that these two systems are now incompatible with each other. So you swap the salt bridge, and now pure-1 is negatively charged, basically, and Hab1 is positively charged. So then this is pure-1 star and Hab1 star. So now pure-1, which we see as positively charged, can no longer bind to Hab1 star, which is also negatively charged. So like magnets, they sort of repel each other, and vice versa. Pure-1 star cannot bind to regular Hab1. And by doing this salt bridge swap, we swap the amino acids that form that salt bridge between the proteins. We can then have this orthogonal system. So we have pure-1 ABA working at the same time as pure-1 star mandipropamide, right? This is really cool and really novel part of this paper. So we have multi-input, multi-output genetic circuits because of this system. Just in many article formats, you have this whole introduction giving background, and towards the end of the introduction you say in this work or here. So in this work, this is actually what we did. We addressed the need for chemically regulated gene regulation systems in non-conventional microbial hosts. So this is a flex, chemically regulated gene expression systems, and further flex, it's in non-conventional hosts. So this is not in yeast that we can easily work with. And so this is sort of the tool that we build, but the surface level demonstration is that we optimize carbon flux to ethyl acetate. So we're pushing more of the cell's energy into ethyl acetate more efficiently, right? So this is the flux that I talk about all the time. So that's all of this paper. That's all we're doing, is we're building this system and then demonstrating this system. A little bit of background. This is the metabolic pathway that we're dealing with here. Very simplified, of course. So you have glucose. I don't like how this figure is laid out. This is the chair conformation of glucose for some reason. Undergoes glycolysis, and eventually you get to this pyruvate molecule. So pyruvate is this node of central metabolism. So you have this network of metabolism, and it's just a node in the network. It's a central node, though, so it's important for a lot of things. So this pyruvate can go to two distinct places, at least for our purposes today. The pyruvate can go into eventually the TCA cycle, the citric acid cycle, in order to influence cell growth. So if we have more pyruvate going to the citric acid cycle, we're going to get more cell growth. If instead of going to cell growth, this pyruvate can be used to produce other products like ethyl acetate in our case, right? And so in this paper, we found two knobs that we can turn here, right? This PDC1 enzyme, we can upregulate that to increase production of ethyl acetate. And then this PDA1 enzyme, we can downregulate to decrease cell growth, right? So we have these two knobs that we can control now, one with upregulation and one with repression. And so we need to build these upregulation and downregulation systems on these genes. So most of this paper is talking about how this genetic circuit architecture is built, because there's a lot of work optimizing that. So figure B, or panel B, is showing how these systems are going to work in the end. This top one is showing the activation system. So we have pure-1 attached to something called the DNA binding domain that sits on the DNA. It sits upstream of the gene that you want to turn on. And then Hab1 is attached to a DNA activation domain. So when abscisic acid is present, it binds to pure-1. Hab1 it comes along, and it brings along this activation domain that turns on gene transcription. So if you add abscisic acid, gene transcription of PDC1 should be turned up. And then we also designed a repression system where instead of having an activation domain, we now are working with this pure-1 star Mandy system and HAB1 star. And so now when we add mandipropamid, a repression domain is pulled along and we repress the PDA1 gene. So these are the two systems that we're going to build in this paper. This is sort of a graphical abstract, what we would call a graphical abstract, which is just sort of an overview of the whole story. So then figure 2 is showing some optimization of the PIR1 and HAB1 pieces in Kluyveromyces marxianus. And this is difficult because you can imagine that if you have a lot of the PIR1 protein or a little bit of the HAB1 protein, you're going to get different activity of this whole system. If you make a bunch of PIR1 and a bunch of HAB1, then the cell is not going to grow very well because it's spending all of its resources making these biosensors, right? And so you want to optimize the expression levels of both of these components in order to get the system to work the best. And so what we settle on here is using a stereotypical high strength promoter, meaning we get a lot of this protein on PIR1 star Mandy. And so this promoter is called TEF1, and then a low strength promoter on the HAB1 star part. So this promoter is called PGK1. And then, the terminators were optimized as well. So we have the CYC terminator and the PGK1T terminator. So normally we put a lowercase p in front of promoters and we put a lowercase t at the end of terminators. So these are well known in yeast, and you can go and find papers that show dozens of promoters and the order of their promoting strength. And this is sort of a static way to change gene regulation in yeast. So this was optimized here, and sort of the best case scenario is shown in panel C, where we get a 38-fold change in fluorescence when we add high amounts of mandipropamid. So we're looking at fluorescence because first, just to demonstrate these systems, we're using GFP, or green fluorescent protein, to be our reporter signal. So the cell will glow green when the reporter is active. So green fluorescent protein, the cell glows green. And all of these other pieces are optimized as well. So there's a part where the DNA binding domain binds. We have to optimize what the DNA binding domain is. We have to optimize what the activation domain is. So there's the Z4 binding domain, which I believe is from the HIV virus or something, or maybe VP64 is from the HIV virus, but we get these activation and DNA binding domains from interesting places, which is kind of cool. And then, so anyway, we optimized this whole system, the reporting circuitry, as well as PIR1 and HAB1 star, and now we have the system where if we don't add any mandipropamid, we get a low signal, and if we add a lot of mandipropamid, so 10 to the 2 is 100 micromolar of mandipropamid, we get a 38-fold change. And then we have this linear range of the sensor as well between, what would this be, 0.01 micromolar up to almost 100 micromolar. We have this linear range to tune that knob, right? So 0.01 to about 100 micromolar, we can tune that knob pretty precisely. So in that figure 2, we're looking at just moving the PIR1 star HAB1 star system from Saccharomyces cerevisiae into Kluyveromyces marxianus, and now we're looking at building that star system as a repression system in K. marxianus. And so we need to find the proper repression domains. We need to re-optimize some of the promoters and terminators and the locations of these things. We settle on this top 1 repressor domain. And then finally, we have this repression circuit where if we don't add any mandipropamid, now we get high expression of GFP. And if we add a lot of mandipropamid, then we get low expression of GFP. So this is cool. Now we have this repression system. And then finally, in panel D, we're looking at the full circuitry of the activation and the repression circuit. So again, all of the PIR1 and HAB1 pieces needed to be optimized. On the left, we have the activation. On the right, we have the repression circuit. For the activation circuit, we're using GFP still. And for the repression circuit, we're looking at M-cherry, which fluoresces red. So it's a red fluorescent protein. And now the really cool part, this orthogonality that we're talking about, we can use both of these knobs at the same time. So As we add more ABA or mandipropamide, then we get repression in the case of mandipropamide and activation in the case of ABA. So we're having both of those happen at the same time, which is great. But still, we're just demonstrating that these sensors work in K. marxianus. So we need to then move them as tunable parts of a metabolic pathway. And again, we're looking at PDC1 and PDA1 expression. So for the activation circuit, we swap out GFP for PDC1, which should push ethyl acetate production. And then for the repression circuit, we swap M-cherry for PDA1, and that should allow us to turn down cell growth. So we can have sort of dynamic regulation by at the beginning of the culture, by the beginning of the growth of the culture, we add different concentrations of mandipropamide and abscisic acid, and we already get significant improvements in ethyl acetate production. So here in the column, we're seeing different concentrations of mandipropamide being added in orders of magnitude. So no mandipropamide, 0.1 micromolar and 1 micromolar. And then for ABA, we're adding 0, 1, or 10 micromolar. There's also this bit about ethanol, but it's a little off topic, so we can just focus on the ethyl acetate on the left side. And so you can see that when we add 0.1 micromolar of mandipropamide and 10 micromolar of abscisic acid, we get about 60 milligrams per liter per OD of ethyl acetate, which is almost maybe twice as much as not adding any mandipropamide or abscisic acid. So we're about doubling the production of ethyl acetate by tuning these parts of the metabolic pathway, which is really, really, really cool. As a side note, this unit of milligrams per liter per OD, the per OD is referring to how many cells are present in the solution. So OD stands for optical density. It's basically how much light can make it through a culture. Typically, we can grow these cells to an OD of about 50. So if you have 100 milligrams per liter per OD at an OD of 50, you're looking at about 5 grams per liter. And generally for industrial applications, gram per liter change, gram per liter production of a product is usually industrially relevant, usually economically viable once you're at that gram per liter range. So these are potentially industrially relevant titers depending on the value of ethyl acetate. So then finally, we can tune the knobs even further by looking at, sort of broken down concentration. So if we add exactly 5.36 micromolar of ABA and 0.35 micromolar of mandipropamide, we now get about 125 milligrams per liter per OD of ethyl acetate. So this is a heat map, one of my favorite kinds of figures. And basically the darker areas indicate more production of your chemical of interest in this case. So, the ABA is upregulating PDC1 expression. So we have our PDC1 expression turned up pretty high to get more ethyl acetate, and our PDA1 expression, so the growth gene controlling expression, the gene controlling growth is turned down just a little bit because we're just adding a little bit of mandipropamide. So we're slowing growth a tiny bit and we're increasing product formation a good amount, right? So these are the knobs that we're tuning. And then there's this bit here, panel A and B of figure 5, where we knocked out a couple of genes that sort of sap flux away from ethyl acetate production. They're called ACS1 and 2. And basically, titers increased by maybe 10-20% by doing this. And then the sort of most interesting part here is the true dynamic regulation of these genes. So if we change the regulation at the beginning of the growth, that's useful because instead of swapping out promoters and terminators and optimizing the gene expression, genetically, which would take a really long time, we can just do an experiment all at once like that heat map and try all different levels of expression really, really, really quickly, which is great. But we can also update these expression levels live. So as the culture is growing, we can change the expression of these genes. So this is what figure 6 is telling us, that in this top graph, if you add mandipropamid and abscisic acid at these optimized concentrations at the beginning of the growth, you get a peak ethyl acetate titer of about 125 milligrams per liter. If you don't add any ABA or MANDI, then you get about 100 milligrams per liter per OD. But if halfway through the growth, so once the cells start growing, then you add the ABA and the MANDI, you then get over 150 milligrams per liter per OD. And that's what panel B is showing too, is that if you don't add any chemical, you get lower production over here. And then if you add the chemicals to induce the genes or repress the genes at the beginning, then you get slightly more. But then if you add them at a particular time, so halfway through the growth in this case, then you get the most production. So you have dynamic regulation, so you can quickly change the expression of these genes whenever you want, but also throughout time you can change the expression of these genes. So this is the really freaking cool part here, is that we have very specific ways to manipulate these genes as the yeast is growing. Very cool stuff. As a final sort of note here, we submitted this paper for review. We got really great reviews back. The only big comment was, are ABA and MANDI toxic to the cells? And so this is the only figure that I actually contributed data to. That's why I'm one of the last authors in this list. And so you can see here, if we don't add any chemicals, so DMSO is dimethyl sulfoxide. It's the solvent that we put our ABA and MANDI in. If we don't add any chemical, the cells in this serial dilution grow fine. And then if we add ABA, MANDI, or both, even at high concentrations in this agar, the cells grow about the same. And so the reviewer was concerned about the toxicity of these compounds, and we said, nope, there's no toxicity of these compounds to Kluyveromyces marxianus. So this is in the supplemental data if you're ever looking at this paper. That's all I have for everybody today. I'm really excited about this paper, and I think it's a demonstration of a really, really cool tool and how far we are along in synthetic biology. And hopefully there's even more applications of this as we move into the future. So if you find this kind of paper interesting, or these kinds of videos, make sure you like the video, subscribe to the channel. If you want to recommend something for me to review next, leave it in the comments below. Thank you so much for watching. Bye.